Test, 1, 2, 1, 2, test.
All right. Good afternoon, everybody, and thank you for coming to another seminar. We have to use the mic now because apparently this is that can be watched. This seminar can be watched live. We're in YouTube and all that. So anyway, well, that's kind of intimidating. Hi, mommy. So anyway, it's great to have Richard Zimmerman with us today. It took us about what 2 years to try to find a date for him to come to the Sea Lab for the second time. He was here 20 years ago. And so he came down to New Orleans for the Ocean Science meeting, and we thought it would be a good opportunity for him to come to the Sea Lab and tell us about his research. So here he is with us today after 2 years in the making. It's really a privilege for us to have Dick with us. He's definitely one of the world's leaders in the field of marine plant ecophysiology, particularly looking at the areas of biooptics and the regulation of photosynthesis. He's done a lot of work. I mean, you look at his webpage, the publications, and it's a lot of quantity and quality, pretty much trying to apply rather sophisticated, but not that difficult to use, methods and modeling to understand, what controls the ecophysiology of marine macrophytes, particularly seagrasses, looking at light, CO2, temperature, and that research has a lot of implications for climate change. And you can see that in the impact that his publications have had. So anyway, it's really good to have him with us these days, and today and tomorrow. If you want to talk to him, please let me know. There are plenty of slots, and tell him what we do and get good insights from a very capable brain. And anyway, again, many accolades, many papers, plenty of grants and awards. But I'll stop there and welcome him and thank you for coming.
All right. Well, thanks again for inviting me here. As Drew said, we've been trying to find a mutually convenient time for both of us to get me down here. And it just so happened that the Ocean Sciences meeting in New Orleans was a perfect opportunity for it. I've been there for the last 3 days, and I can tell you, being surrounded by 7,000 posters and presentations and everything else, it's wonderful to be out of there for at least most of the day, where it's my turn to talk about my bad slides instead of looking at everybody else's bad slides for hours and hours. So I'd like to keep this as informal as possible. So as I'm going along, if you have any questions or anything like that, just raise your hand and feel free to interrupt me. What I'm going to tell you about today is a story of some of our seagrass work that I've been working on for actually quite a long time, but we've made significant progress in the last few years thanks to funding agencies, particularly NSF and Virginia Sea Grant, that have played big roles in our operation. And most of the work I'm going to be telling you about today was done by all of the other people that you see here. Victoria Hill is my co-PI on the project. Molly Januntia and Bill Chelaby are 2 PhD students in my lab that I hope are at this moment are back home in Virginia writing their dissertations. That's what they're supposed to be doing. David Rubel is our marine technician, and he's the guy who keeps all of the instruments and machines and running and keeps the data flowing. Carmen Zayas is a recent PhD student entering our lab. She's starting to look at population level differences in the response of these plants. So that's a new angle that we're taking to this, is looking at a population genetic approach to the responses. So what you're going to be seeing here are responses from a population, but there are implications that different populations from different locations of eelgrass behave differently to these environmental responses. And that's something we're going to be wanting to work with as well. And then we had some very talented high school interns working with us for a couple of years on the project as well. They got great opportunities out of it. And both of these students are now matriculated. Miranda's a computer science major at ODU, and Tiffany Cedeno is in the undergraduate program at UC Santa Barbara, and she's actually working with the Santa Barbara Coastal LTER folks and basically springboarding off of the kinds of things we were able to expose her to here. And I'd also like to make a big acknowledgement to Mark Swingle, our other PI on this project. He's the research director at the Virginia Aquarium, and without the Virginia Aquarium's participation in this activity, our research wouldn't have been possible. They provided most of the facilities. They provided the space for this facility and kept our seawater system running, because ODU, where I work, is an urban campus in an urban environment, and we don't have access to the running seawater that made the experiments I'm going to talk about possible. Okay, so probably can go through this really quickly because all of you know all about seagrasses already, but just a quick review. The important differences between seagrasses and most other marine autotrophs is that they are marine angiosperms. They are true higher plants. That means they have leaves, roots, and rhizomes, and this anatomy and physiology makes a big difference in how they respond to the marine environment. However, they are adapted for a submerged aquatic growth, which means they have unique adaptations and features that allow them to survive in this environment. And in many respects, these adaptations are very similar to convergent solutions that macroalgae have made to survival in a submerged environment. They have no cuticle. They have no stomates. They take up CO2 and nutrients directly across the leaves, and they release oxygen back to the water. The chloroplasts in the leaves have migrated from the mesophyll terrestrial plant leaves out to the outer edge. The roots have air channels that run continuously into the leaves, and so they're supplied with oxygen from photosynthesis by the leaves. All of these are evolutionary adaptations for a submerged existence that allow these plants to survive. So if you look at that, you see a lot of evidence that there's been a lot of evolutionary pressure on these plants to adapt to the marine environment. That's going to be an important theme in this story. Okay, there's more than 50 species worldwide. Some people you talk to will say there's more than 70. They're distributed from polar to tropical environments. They cover more than 600,000 square kilometers globally, which may seem like a lot until you realize that this represents about 0.04% of the coastal, estuarine, marsh, and subtidal habitat around the world. So this seems like a big number in those units, but globally, they represent a very, very small fraction of the ocean. So their global impact on biogeochemistry is even more amplified by the fact that they don't occupy very much space in the ocean. They're just really marginal ribbon around the ocean surface, yet they have an important impact. And that's because they're highly productive. They also create structure and habitat for a diverse array of organisms that all of you are working on, I'm sure. And another interest of mine is that they represent significant biogeochemical agents. They control sediment redox potentials in the sediment where the roots and rhizomes are intimately in contact with the soil. They also are responsible for regulating carbonate dissolution and pyrite formation, which controls the alkalinity in the pore waters and therefore the overlying source waters. And as a result of all this, and as you can see here, they bury a lot of organic carbon in the sediment, so-called blue carbon. So they contribute to carbon burial in a variety of ways, which makes them important biogeochemical agents. And then we all know, because that's all the great stuff, now we all know all the bad stuff. One of the unusual things about seagrasses is that they have remarkably high light requirements for aquatic photosynthesizers. Typically, we define the euphotic zone as the 1% light level in the ocean, yet seagrasses, most places, can barely survive down to the 10% light level, and very often in turbid coastal waters, they require as much as 20% of surface irradiance. So they have very high light requirements. And of course, this makes them vulnerable to all kinds of insults and impacts on water quality, including eutrophication, sediment loading, other processes that degrade the water quality environment hit the seagrasses first. So, and a lot of people have argued that as a result, seagrasses are the canary in the mine of the estuary because they're the first and most heavily impacted. Unfortunately, they're really not the canary in the mine. They're the miners in the mine, because the idea of the canary is the canary dies, the miners get out when they see the problem. But here, by the time the seagrasses die, it's too late, because now you're taking out all the rest of the ecosystem because they form the basis of the ecosystem. So they're more than just the canaries. They are the mine itself. And furthermore, they're sensitive to high summer water temperatures. As a result of all this, seagrass loss threatens provision of major ecosystem services in shallow coastal environments throughout most of the world. And this affects habitat structure and stability. Loss of seagrass means loss of blue carbon deposits, and results in a productivity shift from the benthos into the plankton, which results in significant changes in the nature of the habitat. Rather than promoting groundfish, crabs, shrimp, and things like that, you promote pelagic a food chain, which leads to a completely different ecological structure, not just on the benthos, but also in the overlying water column. And it shifts sediment biogeochemistry processes, it reduces the flux of organic carbon and oxygen into the sediments, changes the redox state, and that affects not only burial of organic carbon, but also the organisms that burrow and live through the sediments. So the loss of seagrasses have these cascading ecosystem impacts that can actually be tremendous. All right, so what I want to talk about today is showing you some experiments that are going to address the issue of the impacts of climate change on seagrass populations in general, with a specific focus on the eelgrass populations in Virginia. So what do we know about climate change and seagrasses? Well, there's a lot of evidence that climate warming is going to increase summertime stress in these populations, not just in the Chesapeake Bay where I live, but throughout much of the coastal environment, where seagrasses are going to experience a significant rise in summertime water temperatures that may cause serious stress. Furthermore, these heat stress events are probably going to become more frequent and of longer duration. And it's not just the average temperature that's rising, it's the frequency of these extreme temperature events that last for periods of 2 to 3 weeks that can become very stressful to plant populations. And then there's this problem of ocean acidification that you've probably all heard a lot about. And we know that there are going to be lots of losers. So anything that needs to lay down calcium carbonate skeletons is going to have a harder time in an acidified environment. But if you look at the chemistry of seawater, you realize pretty quickly that it's more than just pH and aragonite saturation state, which is denoted by this omega here. There are also going to be some winners in this process, because in addition to changing the pH of the water when you add CO2, you also increase the primary substrate for photosynthesis, which means most photosynthetic organisms are going to have an easier time extracting CO2 from an acidified ocean than they are from the present-day ocean. So this could have potential benefits to a number of species, and particularly to seagrasses, as you're going to see in the next few slides. So I would argue that we really ought to call this process ocean carbonation, because it's more than just an acidifying process. And there are going to be winners, there are going to be losers, and part of our role as scientists is to try to understand what the impacts are going to be if we're going to project things into the future. We really want to know how these things are going to sort out. Okay, so back to the seagrass story. We've been doing a lot of work on the role of carbon dioxide in regulating seagrass photosynthesis. And I'll try to go through a couple of these things pretty quickly to get to the other main points. But essentially, if you measure the light-saturated photosynthesis, which I call PE in this plot, relative to the maximum rate of light-saturated and substrate-saturated photosynthesis, you find that there is a strong relationship between this relative photosynthetic rate, and the CO2 concentration in the water. The more CO2 you give the plants, the more photosynthesis you'll get out of them. And this is a curve for eelgrass, but you can do the same kind of thing for thalassia, you can do the same kind of thing for Syringodium, and you get essentially the same curve. What this means is that in the present-day ocean, their photosynthetic rates are around 0.1, And if you give them CO2, you can increase their photosynthetic rates at least a twofold and sometimes as much as fivefold over present-day conditions. And this is an instantaneous response to the CO2 availability in the water. You can do this experiment on a leaf segment that you cut off of the plant, stick it in an oxygen electrode, and just bubble the water in the electrode chamber with a little bit of CO2, drive down the pH, and this will respond instantaneously. What that means is they have all of the physiological mechanisms to do photosynthesis at a much, much higher level than they are presently able to. They simply lack the substrate. They have all the light harvesting capacity. They have all the electron transport capacity. They have all the sugar formation capacity. Everything is there ready to go to work. They just don't have the substrate. And that's part of the story we're going to be talking about today. Furthermore, as a result of some modeling and some experimental work, we can actually break this curve down, which represents the response to the total CO2 concentration, CO2 and bicarbonate concentration in the water, both of which can be used as a substrate for photosynthesis, into their component parts. And so you see the flat line here, this dotted line, represents the response to bicarbonate alone. And what that's telling us is that it's not simply a matter of giving them more dissolved inorganic carbon, because if you add sodium bicarbonate to the water, you don't see an increase in the photosynthetic rate. However, if you add dissolved aqueous CO2, if you bubble CO2 into the water, either from sublimating dry ice or from your tank of CO2, you get a dramatic increase in the photosynthetic response. So that tells us two things. It tells us, yes, they can use bicarbonate for photosynthesis, but their capacity to extract bicarbonate from the water is insufficient to satisfy their photosynthetic capacity. And so as a result of that, in the modern day ocean, seagrasses are carbon limited in terms of their photosynthetic potential, which means they are working at a much lower level of activity than they are physiologically capable of. Well, these kind of experiments allow us to make some simple models to predict the impact of CO2 availability on photosynthesis. And this is just a series of Michaelis-Menten type terms where we can calculate the light-saturated photosynthesis as a function of the maximum rate based on bicarbonate and the good old Michaelis-Menten relationship, plus the maximum rate of photosynthesis given as a function of CO2 aqueous, dissolved CO2, which are basically these 2 points here and here. Okay, these are the Pmax values for bicarbonate and for CO2. And this allows us to look at the photosynthesis rate as a function of CO2, which we can then use to and combine that with temperature, so we can actually get a temperature response to this as well as a result of some other experiments that I'm not going to show you today. But basically, what this amounts to is that the Q10 for photosynthesis in these plants is a complex function of not only temperature, but also the CO2 concentration in the water. If you're CO2 limited and you turn up the temperature, you can't increase the photosynthesis correspondingly to the temperature increase because they don't have a substrate. On the other hand, respiration, which is not substrate limited, just keeps cranking up. So what happens in the seagrasses as you increase temperature is you prevent photosynthesis from rising rapidly enough to compensate for the respiratory demand of the plants, and eventually respiration rates exceed photosynthesis rates and the plants can't survive. Okay, so that allows us to take these equations, put them into a little model where we can actually predict how much photosynthesis would be required for the plants to survive for a range of temperature regimes and CO2 regimes. I'll take you through this plot a little bit slowly. First thing I'd like you to notice is that temperature is increasing along this axis away from the origin. CO2 concentrations, however, are decreasing, and this is simply to make the plot easily visible to you. And on this axis, what I'm plotting here is the number of hours of Pmax photosynthesis or light-saturated photosynthesis required to balance the respiratory demand for a typical seagrass plant. The point of all this is, if we're down here, present-day seawater pH levels, even at temperatures of 5 to 10 degrees, plants need about 4.5 to 5 hours of light at saturating levels to satisfy their metabolic demand. You heat the water up to 30 degrees, and that requirement goes up to about 10 hours. Okay? If we increase the CO2 if we increase the CO2 concentrations, which decreases the pH, you see that the daily HSAT requirements drop dramatically, such that at pH levels predicted for the end of the 21st century, somewhere around 7.8, which is about 1,100 microatmospheres of CO2, we've cut the daily photosynthetic requirement for these plants in half. Okay, so what this is projecting is that sea level or CO2 increases are going to increase the photosynthetic rates, which reduce the costs of maintenance for the plants to survive, which should allow them to survive at higher temperatures, because now, rather than requiring 10 hours of light to meet their metabolic carbon demand, they only need about 5 hours of light. So they're basically cutting it in half. Okay, that's a good thing, because if you get 6 hours of light, that means you're putting money in the bank for use later, for surviving periods, weather periods of low light availability, and more importantly, as you'll see later, to put energy into reproduction in the coming spring. Okay, so CO2 stimulates photosynthesis. This increases the daily ratio of photosynthesis to respiration, and it reduces the effective temperature on light requirements. Okay, now that's based on a model that was derived from experiments done in the laboratory on little sections of leaves cut up and put it in an oxygen electrode, little sections of roots and rhizomes cut up and put it in an oxygen electrode, and then just add up all the components, scale them to the biomass allocation, and make these calculations. Okay, very, very laboratory-oriented reductionist approach. Question is, how does that model behave if we try to do these, try to test that out in the field? Is it really a valid model, and can we use it? So we constructed an experimental facility to test some of these things, and this is where our collaboration with the Virginia Aquarium comes in. We built on their property a climate change test facility that consists of 20 of these fiberglass tanks. They're about 3 cubic meters each, and they're plumbed into the Owls Creek in the background here, which we then feed water into this large storage tank, and then flows directly into the aquaria themselves at a turnover rate of about every 2 hours. So we've got a good flow-through system. This is a single-pass flow-through system. The water goes in at one end of the aquaria, out the other end of the aquaria, and then we can, inside the tanks, we can individually manipulate the temperature, the pH, all kinds of the light availability, all kinds of things as we want, and we get lots of replication in our experiments because we can put multiple pots in there and do lots of manipulations and replication in the system, and it's really good. We could also control the pH. We have little boxes on the ends. Each of these boxes contain CO2 controllers that we built that actually poises the water in each of the tanks at a different pH level by delivering CO2 to the system, and then we can do the kind of experiments that I'll be talking about here. So we have, yeah, pretty much that's it. And this is the layout of the system. We can randomize the treatments of pH or temperature in here, and then all the environmental data sets are continuously logged. We're measuring PAR at several locations around the experimental facility. Each aquarium is connected to a thermostatically controlled heater, although I'm not going to show you the results of the heating experiments today, and a pH-controlled solenoid valve that allows us to control the pH and or the CO2 concentration in the water. Software control and data logging systems, conductivity sensors, so we have the complete suite of information, and all this is recorded every minute for the entire duration of the experiment. Okay, so we went out to South Bay, one of the coastal lagoons of Virginia, which is quite famous because Robert J. Orth from the Virginia Institute of Marine Sciences has been using this as an experimental restoration site where they have been propagating eelgrass populations there by spreading out seeds. So they actually go out and collect seeds and have been distributing there, and it's been very successful. It's almost 1,000 hectares of seagrass come back now in the last 10 years due to their efforts at distributing the seeds. So we used these populations as our donor source, went out and collected the plants by hand, brought them back into the aquarium, transplanted them into these plexiglass trays with sediment from the habitat, and then set them into our aquaria and grew them for about 18 months. And the whole time we were growing them, we were making monthly measurements of shoot number, shoot size, growth rate, sugar content, pigment content, tissue CN, carbon, nitrogen, sulfur levels, photosynthesis rates, and all the parameters that go into the model. We were seeing how they varied through time in our experimental treatments. And simultaneously, we were measuring, continuously monitoring all of these things. The pH in the system was controlled by the delivery of CO2, which we got from a large compressed tank of CO2 that was delivered to the system. And each tank got its own titrated value with a pH controller in them, so that we were able to get good separation across our treatments. Okay, now because this is an outdoor facility, it imposes another level of natural variability on our experimental system. Solar radiation, for example, during the course of our experiments varied by 5-fold from winter to summer, and our plants were exposed to the natural annual range of sunlight availability. The black symbols here represent the irradiances measured by our PAR sensors, and the red dots actually represent the light levels delivered to the plants, which were shaded to simulate the light levels in the environment from which they were collected. And we do this for two reasons. Number one is to control epiphyte growth in the tanks, because that becomes a real problem, a management problem. Number two, we don't want to expose them to really high light levels, because then the plants could actually get sunburned. So it's trying to reproduce a similar light condition to what they saw in the field, but giving them that nice seasonal variation, so we could look at long-term effects and have some expectation that what we're seeing actually represents the true, more realistic representation of the plant's true response to the field. And then there's variability associated with clouds and amplitudes in daily variation. Important thing to notice here, the amplitudes in daily variation were greatest in the summer, much greater in the summer than they were in the winter. And this is simply due to weather scale events. A lot of clouds coming through knocks the light down. So you could have days with low light availability in the summer that are almost equivalent to the average light availability in the wintertime. For example, in the summer, we got about an average of 8 hours of light-saturated photosynthesis every day. In the winter, it was cut in half. It was about 4 hours. So that was imposed upon the plants in the system. The ambient temperature. Here is the plot of ambient temperature. The black line represents the temperature range over the season. So not surprisingly, it's warmest in the summer, coldest in the winter. Reaches an average high of somewhere between 25 and 30 degrees in the summertime, and an average low of around 5 degrees in the winter. The red dots represent the number of hours each day that the temperature exceeded a well-established temperature threshold for stress in eelgrass of 25 degrees C. And so what you can see is during the winter, of course, it's cold, no stress, but during the summer, increased frequency of periods of thermal stress in the system, and for much of July, August, and September, it's almost every single day the temperature exceeds that critical threshold for the entire length of the day. So in fact, in our experiment, the temperature threshold exceeded the stress level for 70 days in 2013 and for 90 days in 2014. So this is giving us a good idea that we should have seen significant temperature stress to the plants in our experimental system during those 2 summers of our experiments. Okay, just briefly, there's also weather scale variability and salinity. However, our pH controllers provide good experimental separation of this. The black line represents the natural ambient pH in the system, and then each of these different lines represents a different pH level or CO2 concentration at which we had poised the plants, and you can see they are all quite separate. The dark lines represent the mean daily pH, and the shaded bars around it represent the maximum and minimum pH recorded during the day. So you see we've got good separation of the pH, and again, because pH is being driven by CO2, this is a change in the CO2 substrate for photosynthesis to the plants. The manipulated treatments were within 0.1 pH unit of the target, and since we know the salinity and the alkalinity of the water, we can actually use CO2 cis to calculate the whole carbonate system and actually even calculate the aragonite saturation state in the water and the systems. And one of the really interesting things you'll see here is that there is a lot of high-frequency variation in the aragonite saturation state in the system, and this is in our control tank, our unmanipulated, the tank that we did not manipulate seawater pH. So this represents the average or the typical daily variation in aragonite saturation state in the creek. So it ranges from much less than 1, which means carbonate should be dissolving, and it should be hard for the oysters to precipitate calcium carbonate, up to periods up to 5 and 6. And on this scale, it looks kind of random in that variability. However, if you take a closer look at that variability, you can see that there is a seasonal pattern to it. The pH tends to be lower in the summer. The average pH tends to be lower in the summer and higher in the winter. There's a strong temperature component to it. And furthermore, if you look at daily variations, you see that there is a very significant diurnal cycle in the pH in this estuary, driven by the daily pattern of photosynthesis and respiration. So in this estuary, the pH range is almost about a half a pH unit on a daily basis, sometimes as much as 1 pH unit. Now, when we think about ocean acidification studies out in the open ocean, we're worried about changes in the average pH of 0.2 units. We're seeing in our estuaries fluctuations in pH that vastly exceed projections for the next century, and we're seeing them in ways that are not necessarily beneficial to the seagrasses because the pH drops. CO2 concentrations increase at night when the lights are off. And then during the daytime, when the lights come on, the algae and everything else are competing with the seagrasses for CO2, and they're raising the pH pretty high. They're stripping the CO2 out of the water, which is having, again, an impact, a potential negative impact on the plants. Okay. So what I want to show you now are some results from those experiments, and I want to go through them kind of quickly. And you're going to see a series of heat maps like this, which all have basically the same pattern. Time will be on this axis, and this will be the duration of our experiment down here. The treatments are represented by these 5 bars across here. And then the responses we have mapped, rather than showing data points, we've contoured them using the averages and the variances across our treatments to give you contour maps of the heat responses. And the values over here represent changes relative to the initial condition. So here we're looking at chute numbers in the tanks. We originally planted the experiments with about 50 chutes per tray in each of those tanks in May. So that represents 100%. And over the 18-month course of our experiment, you could see that treatments that did not get any CO2, at the end of the experiment, we were left with less than 50% of the number of chutes we had originally planted in the tanks. However, as we increase the CO2 concentration, going down this axis, you see the color's getting warmer. That means we're increasing the number of chutes that were surviving. And at this level down here, at about a pH of 6.6, which is not ecologically relevant for climate change, but very physiologically relevant because it shows us that our responses are linear across a very large range of pH. And I'll get back to why that's important in a little bit. You can see not only did our plants all survive, but after the long, hot summer the first year, in the following spring, we actually ended up with twice the number of shoots in the experimental system than we started with. And that's due to vegetative propagation. Okay? It's not due to seed recruitment, because these plants, although they flowered, and I'll show you that in a minute, we're not looking at seedling recruitment. This is all just vegetative growth. In other words, they're just propagating new daughter shoots into the population. So a big effect on shoot numbers, a similar effect on shoot size. Same kind of thing. In the absence of CO2, the plants got smaller. There was a little increase the following spring, but under high CO2 conditions, you see a dramatic increase in plant size all through the first hot summer. And then in the winter, when the light levels go low, everybody gets a little bit smaller. And then as soon as the light comes up, the plants start growing again. And again, these plants with high CO2 concentrations were twice the size of the plants that we had originally brought in from the field, while these plants were up here, the unmanipulated plants, were about the same size they were at the beginning of the experiment. Okay. Absolute growth rates follow that same kind of pattern, an increase in performance with an increase in the CO2 availability. And again, in the wintertime, those effects seem to go away because at low temperature, everything slows down. And then the second summer, you can see even more dramatic effects on absolute growth rates, because here, this is a product of both the increase in growth rate as well as the increase in plant size really takes off in terms of absolute growth rates in these plants. So the effects amplify. Okay. Not surprisingly, sugar concentrations follow the same pattern. If this is all driven by photosynthesis, what we would expect to see is higher sugar concentrations as a result of increasing CO2, which fuels all those metabolic processes. And that's exactly the pattern. Again, all through the hot summer, these guys are depleting their sugar reserves. These guys are accumulating them to levels way beyond what they need for local survival, which means they're putting resources in the bank for use the subsequent spring when the temperatures start to rise and the plants start to grow again. And you see the same kind of pattern. They have the highest sugar concentrations in January and February as the plants get smaller and they're concentrating those reserves. And then when the water warms up, they start utilizing them and growing. Leaf nitrogen content did show just the opposite pattern, in fact. Okay, so this effect of CO2 on the plants resulted actually in a decrease in the relative nitrogen content of the plants. And this is expressed in moles of nitrogen per gram dry weight of the tissue. When we normalize it to plant area, it turns out that the nitrogen content in the leaves is the same. So per unit area, the nitrogen content is the same, but per dry weight, what we're seeing here is that the increased carbon, mostly in the form of sugar, is actually diluting the nitrogen a little bit. That's an important point for grazers who are going to be feeding on this stuff, because now they're going to have to eat a lot more seagrass leaf to get the same amount of nutrition out of the plants. They're going to have to go through a lot more sugar in order to get to the protein that they're after. And just to summarize all of that, often a picture is worth 1,000 words. Well, here you go. So after a long, hot summer, here's what the plants look like. This picture shows the plants performing pH 6.6 in a high CO2 concentration. The leaves were almost a meter long. They spread clear across the tank, and you can see how densely vegetated the trays are. These are the plants that didn't receive a CO2 subsidy, that were simply growing on the natural environment. And this is very typical of the kind of thing that we see in the Chesapeake Bay during the summertime, is this wasting away of the plant performance. And then you can see here differences in the plant size because these plants grow in a relatively constant allometric distribution of leaf, roots, and rhizome. Increase in above ground biomass means a corresponding increase in the below ground biomass. So as we make them bigger, we make them more metabolically active. We're also pushing more carbon below ground, increasing the blue carbon value of the plants as well. Okay, now most dramatically of all is the effect we had on flowering production the following spring. No flowers for the first year. That's typical because we collected only vegetative shoots in that first season. But during the in the subsequent year, what we noticed is not only an increase in the number of flowering shoots, here expressed as a percent of the total number of shoots. So in this case, half of the vegetative shoots were flowering, whereas in the plants that received no CO2 subsidy, it was somewhere around 5%. Now, typically, you go out to an eelgrass meadow, and about 10% of the vegetative shoots are beginning to flower or flower each year. That's sort of consistent with this. What this experiment suggests is that increasing CO2 availability is going to increase the proportion of flowering shoots that we're going to have in the population, which is going to lead to higher seed production. So we're getting a double effect. Not only are we increasing the vegetative response of the plants, we're also increasing the sexual reproductive output of the plants the following year. However, photosynthetic acclimation to CO2 enrichment looks like highlight acclimation in these plants. What we saw in terms of chlorophyll concentration is that chlorophyll concentrations actually decreased during the course of the experiment under high CO2 conditions, okay? Which is exactly what a plant would do if you were comparing a low-light plant, a plant grown in low light, to a plant grown in high light. This is exactly the kind of thing you do. As they increase the photosynthesis rates, you'll see the plants recognize this, and they attempt to downregulate that physiology by reducing the amount of chlorophyll, reducing the amount of light harvesting, because they're catching lots of energy already. But these plants were all grown in the same light environment. So this response that we're seeing here that looks like a high light response is actually a response to CO2 and independent of the light environment. And we see the same kind of pattern with the carotenoids. So this is the chlorophylls, and this is the photoprotective carotenoids as well, which is an interesting response, very interesting response. Okay, so the plants getting high CO2 need less chlorophyll. It also appears that they need less photoprotective carotenoids to defend themselves against photoinhibition. all of these things suggesting that we're stimulating the photosynthetic apparatus by giving them CO2 and making them more robust, and therefore reducing the reliance that they have to make on photoprotective mechanisms and other physiology to prevent themselves from becoming stressed by other processes. And then this is just a product of that. This represents the optical cross-section. It's a measure of chlorophyll use efficiency in the plants. And you can see that the optical cross-section increases with CO2. So they're making more efficient use of the pigments they have. And all of that means that as they get more efficient with all these resources, that means they have to spend less energy on doing those things. They've got more energy to put into other behaviors and physiologies in the plant, making bigger rhizomes and roots, doing other physiological processes that facilitate their survival. Okay, so that leaves us with an important question, and this is something we're looking at addressing next, is why does high CO2 concentration look like high light environment? Well, understanding that involves developing an understanding of chloroplast function, because that's key to regulation of light harvesting complex in all plant systems as we know them. There are a number of ways in which this process is regulated. One of them is through photorespiration. When photosynthesis is limited by CO2 availability, rubisco, the carboxylating enzyme in the chloroplast, undergoes a reverse reaction called photorespiration, in which actually the rubisco substrates combine with oxygen, and they end up burning carbon instead of fixing carbon in this process. This photorespiratory process also stimulates antioxidant responses. So one of the things we think is that by supplying them with CO2, we're reducing the photorespiratory response in the plants. We're relieving them from photorespiration, relieving them from that photooxidative stress, and therefore the plants are seeing this in such a ways, and that could be stimulating changes in chloroplast expression. There are other things that could be going on too in terms of changing reactive oxygen species, which affects the reactivity of photosystem 2 and also stimulates the antioxidant responses that regulate nuclear gene expression, which controls chlorophyll abundance. And then there's always, of course, the redox state of plastoquinone in the electron transport chain of the chloroplast that regulates all these processes as well. So there are a number of points in this cascade of events that control gene expression that ultimately are going to regulate chlorophyll responses in these plants. And we're just now beginning to unravel them, but this is an absolutely fascinating possibility that typically these things are thought to be regulated by light. And now we're showing in our experiments they also appear to be sensitive to the CO2 substrate that the plants are given. So we've got two different pathways to get at these plants and manipulate them. Okay, all of these things here can be affected by the CO2 availability. Okay, so it looks like the long-term experiments support the model calculations. We get despite the fact that they were in the aquarium for 18 months, we saw no downregulation of their photosynthetic capacity, no downregulation in photosynthetic responses, and we saw behaviors in the whole plants that could have been predicted from our initial short-term laboratory experiments. Okay, photosynthesis increases with CO2. The light harvesting acclimation did not reduce the maximum photosynthesis rates, and their tolerance of thermal stress increased with CO2 availability in terms of shoot survival, plant size, absolute growth rate, carbon reserves, and the question we're left with now is, can these results predict the natural eelgrass distributions in the nature? So can we take our laboratory experiments, tune the model a little bit, and now apply the model to plants growing in the field? And that's what we've been doing lately, and also evaluate the response of these plants to climate change. So we've been working with a bio-optical model of seagrass photophysiology that we call Grasslight, which, by the way, is freely available to anybody who wants a copy and wants to play with the model. I'm happy to share it with you, and we can talk about that later. Basically, it takes Chuck Gallegos' water quality model to calculate the diffuse attenuation coefficients and irradiances down through the water column as functions of the chlorophyll concentration, suspended matter concentration, and CDOM. You don't need to have sophisticated bio-optical properties to parameterize these models, and we get really good estimates of attenuation coefficients. which allows us to understand the distribution of light through the water column, not just PAR, but the whole spectrum of light. So we get this is a spectrally resolved model, and then we can impinge upon the seagrass canopy, and then we take that light model and combine it with all of the physiology I've just shown you that's manipulated by CO2 and temperature, and we end up predicting the biomass distribution that could be sustained for a given light environment as a function of depth through the water. And the nice thing about this is we take this relatively complicated model, run it a whole bunch of times, and we end up with a few values that provide a relatively simple relationship, slightly nonlinear relationship between the maximum sustainable biomass, here expressed as leaf area index, as a function of depth in the water. Then what we can do with this, we can combine it with a map of bathymetry, stick it into ArcGIS, and we can map the light-limited distribution of seagrasses across the submarine landscape and predict the density of the plants. So we've been doing this work at a couple locations in Chesapeake Bay. I'm just going to show you one of them at the Goodwin Islands National Estuarine Research Reserve. Picked this spot because there's a long history of seagrass distributions there, and they have water quality data to allow us to drive the model. And this we ran the model for the 2001 year, 2011 year from the VIMS data sets, and this just shows the 2011 distribution of seagrasses around the Goodwin Islands. The blue areas here are a bathymetry map for the area that as you can see, we need to run the model. Okay. So eelgrass at the site are known to be affected by water quality. They're also known to be affected by epiphyte load, periodic thermal stress, and when we run the model for the conditions at Goodwin Islands, you can see we get pretty good coherence between the distribution of the model distribution and that measured, mapped by the VIMS teams from the aerial photographs. However, you'll see that one important difference is that our model provides a lot more textural detail in terms of the biomass distribution than the VIMS maps do, and that's partly a result of the way they collect their data, which I can show you, which I should show you. Let's see. Oh, yeah, I can show you here. Okay. They use aerial photography and supervised classification to define these regions and to find a single value of density for this whole zone. But you can see there's a lot of underlying texture that they're not capturing in their models or in their maps. All right, but in any event, when we take their results, compare them to ours, we find out that we get 95% coherence across the scene where our model predicts the seagrasses should be and the VIMS maps say they are, and about 88% agreement in terms of where we both say they should not be growing. So that's pretty good, I think. If we try to look at density classes, we see that the model doesn't predict nearly as well. We see, at least in the low density classes, we're seeing bigger differences, but as the density increases, we can see that the model predictions compared to the measured values do increase as the density in the populations increase. And you can see part of the reason for that, as I explained before, is that the VIMS maps are not catching the textural details that the model can allow us to predict. So we're now at a point where the model is actually predicting more detail than the field measurements can actually provide for us. So that gave us some confidence that the model is working pretty well. And so then we asked the question, okay, how is temperature and CO2 going to interact to affect the eelgrass distributions at this site? So here's the simulation that I showed you before, which represents a cool temperature, a summer temperature value. We ran the model using 25 degrees C temperature, which is right at the edge of the temperature threshold, and present-day CO2 concentrations, and got this pattern that I showed you before. Again, the darker colors represent denser seagrass meadows. Because we were working with eelgrass, we cut our map distributions off at the zero tide line because we know the eelgrass doesn't like to grow in the intertidal. There's a lot of rupia and other things up in here, but we're not modeling rupia in this context. We're only modeling eelgrass. But this is an arbitrary cutoff that we put into the model. It's nothing inherent in the model itself that says you cannot grow beyond the zero tide line. So the question we want to ask the model is, what happens if we increase the temperature? And, okay, if we increase the temperature, the model does exactly as predicted by field observations, is that you get a dieback in the seagrass populations. Okay, so I'll just flip back and forth, and you can see the dense population's there, heating it up, and it dies back. The edge tends to shallow in. Okay, very consistent with the field observations. Now, if we keep the summer temperatures warm, as in that past simulation, and increase the CO2 concentration, we get the population to go back. So here we've increased it to about 870 microatmospheres of CO2, which represents the CO2, projected CO2 concentration for the end of the 21st century. And if we go 200 years into the future, we get even higher concentrations of seagrasses. So the model is not only consistent with existing present-day distributions of plants at these sites, it also allows us to look forward and project the effects of CO2 on this. So, to conclude then, based on our short-term experiments, our modeling efforts, and our longer-term laboratory experiments, it's pretty clear that CO2 availability modifies the eelgrass response to temperature in terms of sugar content, survival and reproduction, shoot size, growth, below-ground biomass. There are important questions about their ability, how these processes affect other sediment geochemical processes, such as sulfide tolerance and nutrient utilization. In general, our long-term experiments with the whole plants support the short-term responses of the individual leaves, and in fact, we can use the individual leaf measurements to predict carbon budget calculations that are consistent with what we're measuring in the whole plants. We combine the physiology with the bio-optical modeling. We get reasonable responses for eelgrass responses across the submarine landscape. And the point of all this is to suggest that ocean carbonation may help eelgrasses and perhaps other seagrasses survive in a warmer global climate. And I'll stop there. Thank you. Yes? That's a really interesting question, and we have, I didn't show it, but we could talk about it later. We have an epiphyte term in the model as well, so we can actually load epiphytes onto the plants and look at their interactive responses with CO2. Not surprisingly, the more epiphytes you put on the plants, the less photosynthesis the plants do, and the weaker the performances are. But that raises a very interesting question about what's going on in these systems, especially in disturbed areas like Goodwin Islands, where the seagrass populations come and go between years, which means when the seagrass population goes away, so do all the predators. And there have been some really interesting papers published recently, one by Brent Hughes on eelgrass populations in California, and another one by Bob Howarth's group up at Cornell, showing that large predators may be very important in controlling epiphyte growth on the leaves of seagrasses because they prey on the mesograzers, especially the blue crabs and other small fish that eat the amphipods and all the other things that eat the epiphyte leaves. So in the absence of rockfish and otters and other major predators, you've got increased populations of the small stuff that are chomping down on the amphipods that are controlling the epiphyte growth. So it really is sort of a trophic cascade kinds of things happening. And all of these things, of course, are interacting together. In fact, since you asked that question, I'm going to show you just a couple more slides, okay? So where do we go from here? Thank you for asking, by the way. All right. So this is basically all those experiments I showed you basically can be boiled down to this simple Odom's energy circuit diagram. It's just basically boxes and arrows showing that if we understand the CO2, light, and temperature environment, we can pretty much predict the response of the seagrasses to those environmental conditions. We are next very much interested in the sediment geochemical processes, but I'm not going to talk about that. But to get to your question, is we also recognize that the grazers, small grazers, control the epiphytes, which are controlled by small predators and larger predators in this trophic cascade, such that as we remove these things, we increase the power of these predators to take out the small grazers, which affect the epiphyte load, which affects the photosynthesis of the plants. And of course, ultimately, the last step is what is the role of humans in this? And we try to map our impacts at various levels of the ecosystem under these processes, we find out that they touch almost every single pathway and interaction in the whole thing. We're controlling, through our human culture, we're controlling fossil carbon increases in CO2 in the atmosphere, which is affecting the temperature, which is affecting the sediment geochemistry. We're taking out these guys, and some of these guys sometimes, most often these large predators, which is affecting the epiphyte growth. We're also adding nutrients, sediment to the water, which is affecting the life of the plants. So you see the human impacts cascade all the way through this thing. And I think we're getting to a point in understanding a lot of these processes where we can start to put together large coupled biogeochemical models to address these kinds of things. So yeah, I think that was a great segue to open things up. But the interactions and all those interactions at all these levels are really important, and we need to understand them if we're going to really understand how seagrass ecosystems are going to respond to human-induced climate change in the next century, because it's not just temperature and CO2, it's all these other processes. It's sediments, it's nutrients, it's harvesting and exploitation, it's the sediment geochemistry. It all comes together to really describe how these systems are going to respond. Yes, it will. We don't, excuse me, we don't think the optical properties of those materials are going to change a whole lot, although there is some evidence, in fact, that there are some changes in the Chesapeake Bay as the suspended particulate matter may be changing from a higher mineral fraction to a more detrital fraction. We may be seeing some changes in that. But those terms are all in the model, and we can adjust them. And you, depending on what you want to do with the model, yeah, you can go in and manipulate those changes to what you think the relationships are going to be. Or if you just want to increase the chlorophyll concentrations, it's really easy right now in the model to increase the chlorophyll concentrations. If you want to change the optical cross-section of the phytoplankton, yes, that can be done as well. And all of those things you can get at those things in the model.
Does the good guy, bad guy model apply to phytoplankton as well, or is that just the annual?
That's a good question, and there's a lot of work going on right now, and so far the results coming back are somewhat equivocal. It looks like the red algae are going to be more responsive to CO2 increases than the greens and the browns that have higher capacity for extracting bicarbonate from seawater. But it's very much an open question how all of this is going to sort out. And different species, even different species within genera, are responding differently to these different conditions. And every week it seems like there's another paper published where somebody's doing similar kinds of experiments on another plant, and they're seeing either a dramatic response or no response, even to the point of coccolithophores, which you would think would be very vulnerable to ocean acidification because they precipitate calcium carbonate tests. Well, it turns out Barney Balch and others have just published a paper in Science showing that, in fact, not only do laboratory experiments not support that argument, but if you look back through the sediment record in the North Atlantic, high concentrations of coccoliths in the sediment record of the deep ocean are consistent with periods of high CO2 concentrations in the atmosphere, suggesting that there may be large-scale shifts, ecological shifts in the plankton communities that are altering the ecological success of these organisms, even though the physiology suggests that they might have a harder time of it. So what it ends up being is very complicated, lots of nonlinear responses, and everything's coupled by differential equations, and so it's very, very hard to predict from first principle or from simple models what's going on with other things. Seagrasses seem to be much more uniformly responsive to this, and of the 30 species of seagrasses that have been examined so far, there are differences in the amplitude of their response to CO2, but they all show some positive response to increased CO2 availability. I don't know of one instance where seagrasses have been shown to not respond to increased CO2 availability. Even Posidonia in the Mediterranean, photosynthetic rates increase by a factor of 2 if you give them more CO2. John. I think the, the, the calcareous epiphytes are going to have a harder time. That's the first order prediction, and they're going to be replaced by the filamentous algae. So you're going to see probably different shifts in those populations. However, I think it's really important, especially with the epiphytes, to keep in mind that it's not just the physiology out here that's driving it. It's this big box and those recent papers by Brent Hughes and Bob Howarth's group showing the role of large predators on these systems by controlling the medium-sized predators is really what's controlling the epiphyte loads in these populations, even in eutrophic estuaries. So it's a combination of nutrients to stimulate the growth of the epiphytes, and then predatory pressure to keep grazing them down. And it seems to me, or at least from those couple of papers, like these points in the system are having extraordinarily powerful impacts on the epiphyte populations. So I don't know how it's going to happen. I don't know how it will all, it's hard to predict how it will turn out just from understanding this side of the process. We've really got to look at this box as well. No, we did not. In our experiments, the leaf turnover rates were about the same for these eelgrass plants. And they produce a new leaf about every 2 weeks, and the leaves survive for about 8 to 10 weeks. We did not see any evidence that the leaves were surviving longer in our experiments as a result of CO2 concentration. They were simply growing faster. The plants were getting bigger. Yeah, so the biomass turnover rates didn't appear to be changing. And I think that has to do simply with probably a biological clock, a maturation process in the plants. And that may be one of the biggest ways that they avoid epiphyte effects on the plants, is because they're continually throwing away the old foul leaves. Don't put a lot of effort into defending yourself, or the best way to defend yourself against epiphytes is throw the old leaf away and make a new one, rather than trying to armor yourself to prevent the inevitable biofouling community from taking over your biomass. Yeah, well, no, ecologically it's not realistic as a treatment. However, the reason we did it is because it allows us to extend our model calculations beyond a very narrow range that's relevant to climate, but very, very hard to observe physiological changes in the plants because of the statistical variability. The noise overwhelms the signal, and it's very hard to see statistically significant effects in the 100-year climate change scenario. On the other hand, if you take advantage of their ability to manipulate it and stretch the pH range out, and the response is linear over that whole range, then you have confidence using linear regression statistics to analyze the experimental results. Not only can you find significant effects, but now you've got, with the linear regression, now you've got a numerical model that will predict the response in quantitative terms to that change, to an incremental change in the environment however you want it. So it's not just a matter of saying, yes, CO2 has an effect. We can say, not only can we say CO2 has an effect, but it will double the shoot numbers. It will double the flowering production rates. We can actually quantify those things in ways we can put it into the model and use it as a much more quantitative predictor than just asking whether or not there is going to be an effect. So the reason that's the reason we went to that extreme range. We also wanted to find out what the upper limits were on their tolerances of CO2, and it seems to be around pH 6.5. If we push it beyond pH 6.5, the plants at pH 6 did not do quite as well. So in terms of looking at the physiological range of tolerances, that gives us an idea of where we are in terms of mapping the response, and gives us confidence in the model, because now we know if we're looking at 100-year changes, we're well inside that linear response range. Well, yeah, I think so, and especially you realize in the estuary that the pH is changing a lot more than the IPCC projection for the whole ocean. They're predicting 0.2, and probably in our estuary, and I'm sure probably around here, if you stick a pH sensor out there in the water for any length of time, you'll see diurnal changes in the pH in the water that vastly overwhelm the climatological change. And that's happening to the plants on a daily basis as well, which something else to take into account is that there's a lot of variability in the environment that's affecting the physiology of these plants. And so at that level, those low pH values can become realistic. Yeah.